Today, we're diving into an amazing story about how we might defeat superbugs by pairing up ancient plants with some seriously modern science. It's a tale where old-school herbal wisdom meets cutting-edge technology to fight one of the biggest threats we face in medicine. I mean, think about this for a second. What if the solution to our most dangerous infections wasn't in some high-tech lab, but was hiding out there in nature this whole time? It's a wild thought, right? But it's exactly the question driving this incredible new research. And their approach is just brilliant. They are literally taking a traditional medicinal plant, extracting its natural defensive powers, and then using the magic of nanotechnology to forge it into a superweapon against these antibiotic-resistant bacteria. Now, to really get why the solution is so cool, you first have to understand the problem. And in this story, the villain is a superbug that literally builds its own fortress to protect itself. So, meet our villain, MRSA. You've probably heard of it. It's a type of staph bacteria. But this one has, well, it's leveled up. It's evolved to just ignore many of the antibiotics we count on, which makes it a huge cause of dangerous infections all over the world. It's a real and present threat. So how does it protect itself? Well, MRSA is smart. It doesn't just float around unprotected. It constructs this dense, slimy shield around its colonies. Scientists call it a biofilm, but you can just think of it as a fortress. This shield makes it incredibly hard for antibiotics to get in, and that's why these infections are so tough to beat. But of course, every good story needs a hero, and in this case, our hero is a bit unexpected. It's a plant with a long, long history in traditional medicine. This is it, Crotalaria candicans. It's native to India, where it's sometimes called rattlepots. For generations, it's been a staple in traditional medicine precisely for its ability to fight off microbes. And its secret weapon? It's loaded with these natural chemical defenders called flavonoids. They're basically the plant's own built-in army. So you have this ancient plant remedy, and you have this modern superbug problem. The big question is, how on earth do you turn that plant into a high-tech weapon that can fight a 21st century battle? Well, it came down to two key moves. First, the researchers had to find the perfect recipe to create a super potent extract of those flavonoids. Then, and this is the really cool part, they used that natural extract to build microscopic weapons. We're talking about tiny, tiny particles of silver and copper. And here they are. What you're looking at are the actual nanoparticles seen through an electron microscope. They are unbelievably small, perfect little spheres about 1,000 times thinner than a single human hair. You can think of each one as a tiny smart bomb, armed with the plant's power, ready to hunt down and destroy bacteria. So just how much stronger is this new weapon? Well, this table tells the whole story. That MIC value is basically the lowest dose needed to stop the bacteria. For the plain plant extract, you need a dose of 64, but for the copper nanoparticles, you only need a dose of 2. That's a massive jump in power. We're talking a huge upgrade. Okay, so the weapon has been forged, and it's incredibly potent. It was time for the ultimate test. Could these nanoparticles actually tear down MRSA's fortress? So, the researchers unleashed these nanoparticles on that slimy biofilm fortress. This slide here gives you the play-by-play, the before and after, but honestly, the next image is the real proof. You just have to see it for yourself. And here it is. This is the visual proof. On the left, that's the before shot, the dense, thriving bacterial fortress. Now, look at the right. That's after the nanoparticles did their job. The fortress is completely demolished. All that's left are these scattered, vulnerable bacteria. It's a stunning success. Okay, so tearing down the fortress in a lab dish is one thing. That's incredible. But the real test, the one that really matters, is can this weapon actually work inside a living creature where everything is way more complicated? To find out, they used zebrafish. And the results you see on this chart are just staggering. That huge bar on the left, that's the bacterial load in the untreated fish, a ton of infection. Now look at the other two tiny bars for the fish treated with the nanoparticles. The infection is nearly wiped out. That's a massive victory in a living animal. So the weapon works, it's incredibly potent, but that leads us to the final, and maybe the most important question of all. I mean, just let that number sink in. More than a 99% reduction of bacteria inside a living animal. This shows the idea has some serious potential. But when you have a weapon that powerful, you have to ask, is it safe? It's great that it destroys the enemy, but you have to be sure it's not causing any collateral damage to the patient.
And these images gave them the answer. What we're looking at are microscopic slices of zebrafish liver. On the left, you've got a healthy, untreated liver. On the right, a liver from a fish that got the nanoparticle treatment. And they look pretty much identical. The cell structure is normal, there's no damage. This was the final piece of the puzzle. It's not just powerful, it's also safe.
So in the end, this is a story about the amazing things that can happen when you combine the old and the new. It's proof that sometimes the wisdom of the past can inspire the solutions of the future, giving us a brand new and much-needed weapon in the fight against superbugs. And it just leaves you wondering, right? As we face all these new challenges, maybe some of the best blueprints for the future have been hiding in nature all along, just waiting for us to find them.